Hello everyone. In this particular session, I am going to discuss about the electron transport chain. So this electron transport chain is one of the pathway which is linked with the oxidation of glucose metabolism. So far we discussed about the previous steps of the oxidation of glucose, that is glycolysis, citric acid cycle, and after glycolysis and citric acid cycle, now the third process will today going to discuss is electron transport chain. So here we should understand before the two pathways where there will be a synthesis of reduced molecule, that is NADH and FADH to carrier. The electron carrier molecule as generated in the previous step, and those electron carrier molecules are now involved in this particular electron transport chain, where they can donate the electrons in a systematic manner so that it can be generated the proton gradient across the membranes, and finally that can lead to the generation of ATP. So in this particular session, I'm going to speak about a few basics about how these carriers are generated and what are the important organelles that will be involved in this electron transport chain. And of course, I'm also going to talk about the structural organization of mitochondria. This is like an ultra-structural organization. And after that, I'm going to speak about the components of the electron transport chain. These components of the electron transport chain, I'm going to speak about various components and their protein structures and their prosthetic groups and everything I'm going to speak about. And in between, we'll also discuss about the Q cycle, which is to be an important cycle, but I'll try to explain in a better, easy way. And then finally, I'll conclude this topic with the inhibitors of electron transport chain. So I'm exclusively concentrating on only electron transport chain. I'm not going to discuss about the oxidative phosphorylation. That oxidative phosphorylation, which is the final step for the generation of cellular energy, that is ATP, that I am trying to make into the coming class. Now, you just think about this. In a previous two glucose oxidizing pathways, where the glycolysis and Krebs cycle, the net production of ATPs are 2+2, only 4 ATPs are generated in the previous cycle. But these 4 ATPs, if you observe this, these are not enough for the cell to do its various activities. So there is a need of extraction of more energy in these particular intermediates which are formed in this glycolysis and Krebs cycle. So definitely, if you take a best example, so one thing is electron transport chain or oxidative phosphorylation. So this is electron transport chain and oxidative phosphorylation. These are the pathways where that can be extracting maximum amount of energy so that the more amount of ATPs is generated. Here I am going to give you the best example. If you see this, for suppose a working muscle, if you take a working muscle, the working muscle recycles over 100 million ATPs per second. You just think about it. So if a muscle which is to be in working condition, it can take a huge amount of ATPs and that can use it for its activity. So definitely there is an other mechanism other than this substrate level phosphorylation reactions of glycolysis and Krebs cycle, so that these electron transport chain and oxidative phosphorylations are the other mechanism from which it can extract maximum amount of energy from the compounds. So if we want to understand this electron transport chain, we should also be pretty much have the knowledge about the mitochondria. So definitely the mitochondria is a major organelle which will be involved in this energy generation pathways, that is electron transport chain and oxidative phosphorylation. So if you see this, the major theory which is to be involved for the generation or origination of mitochondria in a eukaryotic cell is to be endosymbiotic theory. This theory is explained and beautifully how this mitochondria is symbiotically entered into the eukaryotic cell, and finally it has to be present as a cell organelle. But anyway, here I'm not going to be discuss much about the endosymbiotic theory, but you should remember that the origin of the mitochondria and eukaryotic cell is to be explained by the endosymbiotic theory. So if you see here, there will be an if you take the mitochondria and go for the transfer sections, and you should understand the various different parts of the mitochondria. So this is important to understand the functions of the mitochondria. So here you just looking into the various kinds of the groups, and I'm taking the annotation for a better understanding. So if you take the mitochondria will be a double membranous organelle. So double membranous organelle, where this part we call the outer membrane, and there will be one more membrane that can present in the inside, we call inner membrane. So this outer membrane and inner membrane together will make the mitochondria will be double membranous structure. And if you see here, there will be space between between these two membranes. We call this space is intermembranal space, or we call them as an intermembranal space. This intermembranal space is important for generation of the proton gradient. That is what is this proton gradient and how it is to be important, that we will see the upcoming slides. And you can also see here, there will be a space that can be present in the inner mitochondria. We call them as, this is matrix. So matrix is nothing but that is the core of the inner mitochondrial membrane, we call matrix. And there is a projections that can present or that can originated in the inner mitochondrial membrane. These projections we call cristae. So these are many different projections that can be protruding into the matrix, and they can form like satellite structures and villi-like structures. These we call cristae. And this cristae, the advantage of cristae is to be having an increasing of surface area, so that can be accommodated many different kinds of the proteins and enzymes, which will play the various kinds of metabolic role. And anyway, here I am not going to discuss what different kinds of the enzymes or what different kinds of the proteins which are present in a cristae. But you should remember that this is the cristae or nothing but these are the invasions of the inner membrane of the mitochondria. And obviously this mitochondria, we also call it is a self-autonomous organelle, because that can have the chromosome which is to be circular DNA. The circular DNA that can present in a mitochondria, and that can make the proteins which are involved for various functions of this mitochondria. And this can be involving in various kinds of the reactions, and that anyway we will see in the coming slide. So we should better understand about what is the importance of this mitochondria, because the importance of mitochondria is to be like this. So if you take mitochondrial matrix, as I explained, the matrix is nothing but that can be present inside of the when you're going from outside to the inside, first outer membrane and inner membrane, and then in the core is to be matrix. The matrix is rich with many different kind of enzymes. Here I have given one example is pyruvate dehydrogenase complex. As I explained in a citric acid cycle, this pyruvate dehydrogenase complex is involved for the conversion of pyruvate to the acetyl coenzyme A. So this acetyl coenzyme A is to be a substrate for the citric acid cycle, and all the enzymes which are involved for the citric acid cycle, totally 8 enzymes that can be present in the matrix of the mitochondria. And not only that, there are few other enzymes like fatty acid oxidation pathway. This we call beta oxidation, and even the amino acid oxidation enzymes, these can also be present in the mitochondrial matrix. In a one word, we want to say that all pathways of the all pathways for the giving of oxidation or for giving of energy that can be involved, that can be present in the matrix. And the except one pathway, that is nothing but glycolysis. If you remember, in glycolysis is to be taking place at the cytoplasm of the cell, not in a mitochondria. But rest of all other pathways like the conversion of like the pyruvate dehydrogenase complex and citric acid cycle and beta oxidation pathway and amino acid oxidation pathways, all are present in a mitochondrial matrix. So if you see the structural importance of these two membranes of mitochondria, where if you take first membrane, that is outer mitochondrial membrane. So this outer mitochondrial membrane, this is readily permeable to the small molecules and ions. So this is possible because of there is a special kind of channels that is present, we call mitochondrial porins, and also we call voltage dependent anion channels. These VDACs can present in the outer membrane of mitochondria so that it can be freely transport the small molecules and ions across the outer membrane of mitochondria. But if you see the inner membrane, it does not have these VDACs, so that it is impermeable to most of the small molecules and even ions and even protons. This is important, so that the proton gradient that is to be accumulating in the intermembranal space, that is the space between the outer membrane and inner membrane, the intermembranal space will be accumulating the proton gradient. That is because of various electron transport components. So this proton gradient will not easily diffuse through the inner membrane, and which is not to be coming to the matrix. So there will be an other enzyme that can be involved for this transfer of this proton from the space to the matrix. So that's how the ATP is synthesized. But anyway, here the pathway is called oxidative phosphorylation pathway. Today I am not going to discuss that. Today I will discuss how this proton gradient is formed in the membranal space. So before going into the actual electron transport chain, we should understand about the basics about the electron carriers. This is important. So what are electron carriers? If you see the glycolysis, Krebs cycle, these two pathways, exclusively we found two electron carriers. One is NADH and second thing is FADH2. These are the electron carriers which can be taking electrons from these various pathways, from the various substrates, and they are reduced and they are converted into the NADH and FADH2. If you see here in a glycolysis, there is a step that that conversion of glucose 3-phosphate to glucose 3-phosphate to 1,3-bisphosphoglycerate, where the conversion of NAD plus to NADH is taking place. So this is two molecules will be converted, so two NADHs can form in a glycolysis. And if you see the Krebs cycle initially the pyruvate will be converting into acetyl-CoA. So in this particular reaction, each reaction will generate one NADH. So this one NADH is for one molecule of the pyruvate. So here two molecules of pyruvate, so two molecules of NADH is produced. So if you see the entire citric acid cycle, 3 NADHs are produced. So two molecules, one acetyl-CoA will be involved in citric acid cycle and produce 3 NADH, and total 2 acetyl-CoAs are produced. So definitely 6 NADHs are produced. 6 plus 2, total 8 NADHs are produced in the citric acid cycle, and 1 FADH2 for one cycle of the acetyl-CoA. So total 2 FADHs are produced. Now you should remember that 2 NADHs in glycolysis 8 NADHs in citric acid cycle, and 2 FADHs are produced. And these NADH and FADH2s, we call them as, these are the electron banks. So electron banks mean that can again will ready to donate its electrons in the electron transport chain, and that will be important for the this process, that is electron transport chain. There is a systematic taking of these electrons from these respective electron banks, and that is to be important for creation of the proton gradient in the membranal space. So now coming to the actual electron transport chain. So here we should understand the few basics, because if you are, for suppose these electrons, if you consider these are the different steps at the top, which is to be high energy electrons that is presented, and these electrons when going down, and it can be converting into low energy, and during which can release certain amount of energy, and this energy will be utilized for the production of ATP. So here you should understand that each and every complex will be taking electrons, and that is giving back to the giving to the step by step, which is finally going into the down. So here you should understand this, the carrier, this is nothing but carrier to carrier transfer. So one carrier to other carrier. So if you always compare it, when you are going down, definitely the electronegativity will be increased. What do you mean by electronegativity? Electronegativity is nothing but the attraction of the electrons will be more. When you are going down, the attraction will be more, so that the electrons are slowly transferred from high energy to the low energy. So this is a beautiful diagram where you just imagine, or you just think about how this can be happening. So now coming into the actual components of electron transport chain, where 4 different components that is present in electron transport chain. We call complex 1, 2, 3, and 4. Why we are calling it as complex? Because which is combination of many different type of proteins and enzymes, even prosthetic groups will make a single complex. So that's what we call complexes. Complex 1, 2, 3, 4. And here you can see complex 1 we call NADH dehydrogenase, where this is a major enzyme, or this is a major enzyme that can be present, or we call complex 1 as an dehydrogenase, NADH dehydrogenase, which is involving 46 proteins, and the prosthetic group is FMN and 9 iron sulfur centers. Complex 2 we also call succinate coenzyme Q reductase, where total proteins are 5, where prosthetic groups are FAD, cytochrome B, 560 and 3 iron sulfur centers. Third one is cytochrome C reductase, where 11 proteins, and there will be involvement of cytochrome BH, BL, C1 and iron sulfur center. This we call risky center, which is important for the Q cycle operation. And the final one is cytochrome oxidase, where 13 proteins are involved, and where the cytochrome, different kind of cytochromes like A, A3, and copper center A, copper center B is involved in this particular things. And now I am going to discuss about each and everything, how the electrons are transporting in each and every complex. Now coming to the complex 1, which is NADH dehydrogenase. So this is a complex 1. In this complex 1, how these electrons are taking place, this we should better understand. So NADH, as we discussed, these are the electron carriers. NADH will give the protons, NADH will give the 2 electrons. 2 electrons will be taken by the FMN. This is a prosthetic group, and where it can be again given into iron sulfur center, and from where it can be converting in, it can be again transferring to the iron sulfur center protein, that is N2, and finally from N2 it can be given to the ubiquinone, and this ubiquinone, this can also be take the 2 protons from the matrix. You should remember that this is N side and this is P side. P side is nothing but the intermembranal space, and N side is nothing but the inside of the mitochondria, that is matrix side. So it can take 2 protons from the matrix and 2 electrons from this N2 protein, and finally it is converting into the ubiquinone. So this is a reduced form, QH2. So during this transition of the electrons from NADH to the finally formation of QH2, this can lead to the pumping of the protons from matrix to the inner membrane space has taken place, where total 4 protons are pumped. So this we should remember. And the complex 1, 4 protons are pumped out from the matrix to the inner membranal space. This is to be on first one, that is complex 1. And coming to the second one, complex 2, we call succinate coenzyme Q reductase. So if you see the structure here, this structure will have a 2 transmembrane domains. So better I will take this. This structure will have a 2 transmembrane domains. You can see this one and this one. So these are, we call domain C and domain D. So 2 transmembrane domains, that is C and D, that can be inserted into the inner membrane of the mitochondria, and cytoplasmic domains also be present. There will be 2. This we call cytochrome C. The cytoplasmic domain is A, cytoplasmic domain is B. So this cytoplasmic domain A and B will have the FAD and iron sulfur centers, where exactly the electron transport is happening. Here in a better way I have shown here. So this is succinate dehydrogenase, is an enzyme that found in the inner mitochondrial membrane. This is to be involved in the citric acid cycle. So it's a flavoprotein with an FAD as a coenzyme. So this can accept 2 hydrogen atoms, so that it can 2 hydrogen atom from the succinate, and it can be transferred into the finally coenzyme Q. This will be looks like this. You can see here succinate is giving 2 hydrogens and 2 electrons to the FADH, and it is converted into FADH2, and finally it is giving to finally it can be passing through the electrons through the iron sulfur centers. So there will be 3 iron sulfur centers is possible. This we call center 1, center 2, and center 3, and finally it is given to the coenzyme Q. So this is how, this is only the FADH2 only can enter into the complex 2, but all NADHs are entered in the complex 1. This we should remember. NADH electrons are entered in from the complex 1, and that can directly giving into the coenzyme Q, not complex 2. Here the FADH2 that can be directly giving electrons to complex 2, not complex 1, and complex 2 to directly which can giving into the coenzyme Q. This is the major difference we should understand. And here the important thing is that whenever the electron is transferring in this complex 2, it does not pump any protons. This is important. So previous complex 1, 4 protons are pumped from the matrix to the membrane space, but in a complex 2, there is no proton is pumped from the matrix to the membranal space. This we should understand. And coming to the complex 3, we also call cytochrome C reductase. In cytochrome C reductase, which is important, where exactly the Q cycle is operated. What do you mean by Q cycle? So here we are going into in-depth knowledge of the Q cycle. Here I am trying to explain in a simple manner. So you just see this circle we call as a risky center, or risky center, where exactly that can be hold 1 electron for certain time, and the 1 more electron is passing into the other pathway. Here exactly how it is to be happening, that I'll explain here. So if you take this reduced ubiquinone, that is coenzyme Q, which is giving it 2 electrons. So on 2 electrons, the oxidation of QH2 will be taking place in the 2 parts. This is to be on first part. This part will be on first part, and this is to be on second part. So here the first part, what is happening? It is taking the electrons. The electrons are transferring into the iron sulfur center, and from where to cytochrome C1 and cytochrome C1 to the cytochrome C, and where it can stay some time. And one more electron which is to be passing through the heme group, where heme prosthetic group, which is to be having with the BL and BH, and it can be converting into the oxidized ubiquinone. So that is dot indicates which is oxidized state. And this is the first half of the reaction. And second half, exactly what is happened, one more electron is formed, and which is again to be going back, going to the heme center, BL and BH, and which is giving to the oxidized oxidized ubiquinone, and this is converting into the reduced form. So here finally the ubiquinone is nothing but which is to be regenerated. This particular cycle we call as Q cycle, the regeneration of Q cycle. This is possible that at a time only one single electron is to be passing from the these iron sulfur centers, and one more electron will be through the other center, that is a risky center. So this is how the Q cycle is operative. For the better understanding, I have given a few more important points about the Q cycle. So here the few important points about the Q cycle we need to understand. That is, what do you mean by Q cycle? Q cycle is nothing but electrons are transferred from the ubiquinol to the cytochrome C, is known as Q cycle. In the first, there will be two cycles that can be present. We call first half cycle and second half cycle. In the first half cycle, ubiquinol molecule attaches into complex 3 and transfer the 2 electrons into the complex. One of these electrons move on to the risky center, then on to the cytochrome C, C1, and finally cytochrome C. One electron going into this risky center. And note that cytochrome C, unlike ubiquinone, can only carry a single electron at a given time. That's what which is to be the passing of these 2 electrons will be on different pathways. One is to be going back, one is to be going to the risky center where cytochrome C1 and C is to be present. And one more thing, it can coming into the heme group where the cytochrome heme group is nothing but BL and BH, where the cytochrome B and onto ubiquinone to form the partial reduced species of ubiquinone. So one thing is going like this, the risky center, other pathway is to be heme prosthetic groups like this. So these are to be a few important points we should understand. So you can see here, this is to be one pathway is going to the iron sulfur center, cytochrome C1 and cytochrome C. One more electron is going to be heme BL and BH like this. There will be 2 pathways. Only one electron at a time is to be passed. So this cycle we call Q cycle. And before going into discuss about the 4th complex or 4th component, before that we will discuss about the cytochrome C. The cytochrome C which is a molecule that can be connecting link between complex 3 to complex 4. So that will take the electrons from the complex 3 and which will move in the transmembrane, which will move in the inner transmembrane, and that can give the electrons to the complex 4. So this is we call a mobile molecule that can be present between the complex 3 and complex 4. So if you see the basic properties of complex cytochrome C, this is a protein with the heme prosthetic groups. They absorb light at a different characteristic wavelengths. According to that, that can be classified into many different types. So especially when come to the electron transport chain, there will be four different cytochromes are possible. So majorly A, B, C. If you see this A type will have the 2 subtypes, that is cytochrome A, cytochrome A3. You see the B cytochrome C, that will be in a B1 and B2 forms. If you see cytochrome C, that will be cytochrome C and C1. These are the 2 different forms of the A, B, C is possible in this electron transport chain. And coming into the last complex, that is cytochrome oxidase. In this cytochrome oxidase, we should remember that this can be involved with the majorly 3 protein subunits we call subunit 1. Better I'll take annotation here also. So the major reactions which will be involved with the help of subunits, which is subunit 1, subunit 2 and subunit 3. So these 3 subunits will majorly involve, and the rest of the green part will be involved with other 10 protein molecules that will be and supporting molecules. Here you should be understand this cytochrome C at a time. 2 cytochromes will be given 2 electrons, and total 4 cytochromes will be giving to 4 electrons. And first initial these 2 electrons will be giving into the complex. This is to the subunit 2, and subunit 2 this is to be an copper center. The copper is to be prosthetic group we call CUA, and from which it is to be transferred into the subunit 1, where this is to be heme center. Initially, which is to be transferred into A and again A3. At this particular stage, the molecular oxygen is to be interacting with this A3. You can see this molecular oxygen which is interacting with the A3 as well as CUB, copper B center, and from this part which is to be oxidizing to be an peroxidized state, and finally it can take the 4 protons from the matrix, and which is finally converting into the 2 molecules of water molecules. So 4 electrons, 4 protons, and which is finally taking the oxygen will converting into the 2 molecules of water. So during this reaction, so 4 protons are pumped from the matrix, pumped out from the matrix to the P side, that is intermembranous space. So here exactly you should remember that this is the pathway where the oxygen is involved. The final acceptor of the electron is to be oxygen. If there is no oxygen, there is no acceptor of these electrons from the complex 4. So there is no electron transport chain. If there is no electron transport chain, there is no proton gradient that is generated in intermembranous space. So if there is no proton gradient, there is no working of ATPase pump, and finally there is no production of ATP. This is how the reactions are dependent. So that's what this is to be an aerobic oxidation of the glucose. If you see the Krebs cycle which is operative only oxidative conditions, where exactly there is no direct involvement of oxygen is taking place. But the involvement of oxygen is here in an electron transport chain. If it is to be clearing the reducing equivalence, then only the oxidized electron carriers will be again participating in the glycolysis and Krebs cycle. Otherwise, if there is a limiting of the oxidizing NAD plus and FADH plus, so definitely the cycle of glycolysis and citric acid cycle will stop. So here you can see the electron transport in the complex 4 is to be like this. Initially the electrons are cytochrome C from where to copper center A, and that is to be transferred into cytochrome A and heme A3 and copper B, and finally that can be taken into the oxygen, and this will convert it into 2 molecules of the water molecule. So altogether you should remember that the entire complexes, if we want to connect it in a single diagram, this looks like this. So here you just see this is complex 1. At exactly complex 1, the electrons are given by the NADH, and this is complex 2, where exactly electrons are given by the FADH2. This is the difference between complex 1 and complex 2. Finally, these complex 1 electrons are giving into coenzyme Q, and even this complex 2 also will give the coenzyme Q. So the connecting link between complex 1 and complex 2 is coenzyme Q that can take the electrons. Either it can come from the complex 2 or it can come from the complex 1. And this can be transferred into complex C, complex 3, where it can be B, cytochrome B, iron sulfur center, and cytochrome C1, and it can finally giving into the cytochrome C. We also call it as cytochrome C is a connecting link between the complex 3 and complex 4. it will take the electrons from complex 3 and that can give the electrons to complex 4, and in a complex 4 the electrons are passing on to cytochrome A and cytochrome A3, and finally it can giving into molecular oxygen, and that can converting into, or that is reducing into reducing, and that form will be water molecule. So this is how exactly which is happening. Here I am not going to discuss about this particular part. This we call oxidative phosphorylation. I'll try to make an I'll make the next video about oxidative phosphorylation, how exactly the ATP is synthesizing. So all these reactions we call, this is to be electron transport chain. So coming to the last slide of this particular presentation, this is nothing but inhibitors. So these inhibitors are important to understand the various pathways or various activities of cell, because if we are well known about inhibitors, this can be targeted for the drug therapy, and which is to be important for many medical applications, so that it is better to be understand about the inhibitors of electron transport chain. So you can see, I'll take the annotation here, and you can see here, this is the complex 1. Complex 1, the inhibitors are amytol, retinone, and pyridine A. These amytol, retinone, and pyridine A, this can inhibit the cytochrome, this can inhibit the electron transfer from the iron sulfur center to the ubiquinone. So this is iron sulfur center to the ubiquinone. This is act as the complex 1. And at the complex 3, the antimycin A and ball, this is to be involved at the complex 3, and that can be block the electrons transfer from cytochrome B to cytochrome C1. This ball we also call is a British anti-leukosite, and this is to be important for the blocking of complex 3. And even this cytochrome C, and that can be further transferring electrons to A3, and finally oxygen. So here this is complex 4. On complex 4, a few molecules like cyanide, carbon monoxide, and sodium azide. These are the 3 molecules that will work on the that will inhibit the complex 4, so that the electrons are not finally transferred to the oxygen, and there is no formation of water molecule. So that can be, we generally call this is inhibition of cytochrome oxidase, that is complex 4. So here the important thing is we should understand the cyanide poisoning. Though cyanide poisoning nothing but even a very minute amount of the these cyanide will be sufficient to block this electron transport chain, and because of this blocking, there is no production of ATP, so there is no activities of cell, so completely the cell will go for dead. So the cyanide poisoning and carbon monoxide and sodium azide will work on the complex 4. Antimycin A ball will work on the complex 3. Amytol, retinone, and pyridine A will work on the complex A. And students, you should remember exactly where this the inhibition is happening in between the transferred molecule. You should be remember for the many entrance point of view, and the rest of the thing now. If you see the entire structure, what I discussed so far is, which is how the systematic transfer of electrons from NADH and FADH to that will be given into the water, and finally it is reducing to water molecule. And during this various transferring process, that can be pumped out the protons from the matrix, mitochondrial matrix, to the inner membranal space, that is intermembranal space, and where the proton gradient is generated in the intermembranal space. And we also discussed about the various inhibitors and where exactly these inhibitors is worked on this various components of the electron transport chain. So the next class we will discuss about the oxidative phosphorylation, and see you in the next class. Bye.